Afternoon, everybody, and welcome back. We are going to continue with some very interesting talks and talk titles this afternoon. As with the morning, I encourage everybody to drop some comments or questions into the chat, and we'll get to them as we are able, depending on the time. And let's see, I think that we have folks in. I guess people will be coming in from the waiting room as we start. But in the interest of time, let's go ahead and get started now. I think we have both of our next speakers ready to go, it looks like. And so it is my pleasure to introduce Hannah Freund and Talyssa Topacio, both PhD students here at UC Riverside, who will be presenting on the microbial connection between dust and health in the Salton Sea. So Hannah and Talyssa, you are on. Thank you.
Thank you. All right, cool. Hannah, are you good? Ready?
I'm ready.
Okay, awesome. So this is going to be a little bit redundant, but thank you, Dr. Porter, for the introduction. My name is Talyssa Topacio.
And my name is Hannah Freund.
And we are going to be presenting on the microbial connection between dust and health in the Salton Sea. Next slide. So just a little bit of an overview of our talk today. We're going to go into the Salton Sea environment and its community, a bit of a project overview of what we're doing in the lab, as well as sharing some current data that we have. And then also we'll be touching on the broader impacts of our work as a whole. So the Salton Sea was once a thriving freshwater environment. However, more recently has come to be better recognized as an ecological crisis or a hypersaline system in decline. And this is the result of major water diversion and steadily increasing temperatures in the region. Next slide. So in our lab, we are interested in how these ecological events have affected the microbes of sub-environments, particularly in the playa or soil, seawater, and the dust. And as you'll see in later slides, the dust is really something we highlight in our lab. However, we want to note that this isn't necessarily the full story. While the dust gives us insight into the environment, it is only a piece of the total system, right? So while we're trying to understand the connection between host and human health and the environment, considering only the dust without looking into its origins does not necessarily address how the dust microbiome comes to be, and therefore which microbes are going to be the most interesting to look at. Next slide. So as I mentioned, in addition to the environmental microbiomes, we also intend to consider the community itself. So the Salton Sea community itself is faced with a public health crisis, particularly characterized by high rates in respiratory diseases. And this will be touched on a little bit more in detail in the next presentation after us, given by Trevor Biddle from the Low Lab. And so we presume in our lab that there might be a microbial level interaction occurring between the host and environment systems that might be exacerbating this high disease rate. So in the context of microbiome specifically, we presume that the lung sits at an interface between the environment and the host, and prolonged inhalation of the dust and its microbial constituents therefore lead to shifts in composition in the host lung microbiome. So we hypothesize that ongoing desiccation of the Salton Sea influences the dust microbial community, and therefore chronic exposure to this dust negatively influences the lung microbiome of local residents. We aim to address this hypothesis by first examining the microbial diversity of Salton Sea submicrobiomes, particularly that of the dust, and also examining the effects of chronic Salton Sea dust exposures on the host lung microbiome.
So now I'm going to finish off by kind of walking you through how we collect samples and our workflow, as well as some data we have to share with you all. So as you can see on this map of the Salton Sea on the right-hand side, these are all of the sites where we are currently collecting dust and playa material. And on the left-hand side of this slide, you can see our dust collectors. We call these passive dust collectors because we leave them out for long periods of time, and they're far above the ground so that we can catch airborne dust rather than settled dust. And the reason for that is because most likely residents nearby are being exposed to airborne dust coming off of the Salton Sea. As you can see, our lab specialist, Mark, is also collecting playa in this picture. And just to reiterate that we collect playa from all of the locations where we currently collect dust. We also work with Dr. Tim Lyons' lab, which they go out to the Salton Sea and collect seawater at varying depths so that we can compare the microbial communities across all of these different, what we like to call sub-ecosystem microbiomes. So this slide is showing you the overall workflow that Talyssa and I are using to analyze these data. So just to kind of briefly walk you through it, we collect samples from the playa, the seawater, and the dust. We extract DNA from these environments, and then we use these dust particulates to expose mice directly to the dust and see how their lung microbiomes are influenced by this dust exposure. We then extract DNA from the lungs and then submit all of these DNA extracts for different types of sequencing. So we use amplicon or marker gene sequencing to determine the taxonomic diversity, both microbially and of fungi within the Salton Sea dust, playa, and seawater. And then we use shotgun metagenome sequencing to not only confirm the taxonomic information we get from the amplicon data, but also to determine the functional diversity from all of these respective microbiomes. So now to share some data with you all. This is a box and whisker plot showing you the microbial phyla within these different sample types from the Salton Sea. Now you're only seeing phyla that have a relative abundance of at least 5% or more. So I would like to call these like the major phyla of these different substrates. And what's interesting here is that you can see, even though there are maybe different proportions of these taxa in the seawater, the soil, and the dust, there are phyla being shared across all these sub-ecosystem microbiomes. And that supports this idea that there's really connectivity between these sub-ecosystem microbiomes. Thus, there's exchange of microbes, metabolites they could produce, and genetic information as well. Sorry, there we go. So now here's some more data focusing kind of at a finer taxonomic scale, looking at microbial genera between Salton seawater and Salton Sea dust. Now, what's interesting about this bar chart is that this is only looking at shared taxa between Salton seawater and Salton Sea dust. So all of the genera listed on this left-hand side are found in the Salton seawater and Salton Sea dust, but they're clearly at varying proportions. Some taxa are far more prevalent in seawater versus dust and vice versa. So again, this reiterates that there is some exchange going on between these sub-ecosystem microbiomes, but we're really not sure to the extent of how these microbes persist and disperse between this entire environment. So we hope to entangle that more as we submit more of these data for sequencing and further analysis. So why does this matter? Well, as Dr. Lowe reiterated in his talk, the Salton Sea crisis is impacting all those living around it, but also the Salton Sea is not a unique situation. We're seeing saline lakes across the globe decline rapidly due to temperatures rising, precipitation declining, and the diversion of water to meet the needs of the public. And all of these are influencing how rapidly these lakes desiccate and thus lead to higher dust emissions throughout the world. And again, as higher dust emissions occur, we will see more and more of these respiratory issues affecting not just those living next to these environments, but far beyond, seeing as dust can travel for thousands of kilometers. So with that, we would just like to thank our PI, Dr. Emma Aronson, the rest of our amazing lab for all of their help and hard work with collecting these samples and processing them and getting these plates submitted for sequencing. We'd also like to thank our collaborators, Dr. Tim Lyons, Dr. David Lowe, and Dr. Will Porter, and all of their lab mates for just being amazing collaborators. And with that, we'll take any questions you may have. Thanks so much.
Thank you so much, Hannah and Talyssa. That's great. I had a quick question for you. I didn't see one in chat, but I had definitely one that I hear a lot in regards to this type of work. And I was hoping that maybe you could chime in on briefly. And that is, one of the common questions we get, of course, is whether we can find, a mechanistic reason for some of the effects that we see, say, in the mouse chamber, as we'll see in just a little bit as well. What are your thoughts on in terms of how much we can connect those dots based on the work that you're doing in terms of identifying specific pathogens or microbial constituents that might be driving the kinds of health outcomes. What are your thoughts in terms of precision and like mechanistic answers in terms of, and I know that it's still preliminary in some cases, what are your thoughts in terms of potential for developing that kind of flow from cause to consequence?
Right, so I think in terms of what we hope to do or what we intend to do with these sequencing endeavors is not only look at, community composition, but Hannah's super interested in functional diversity as well. And so I guess this is going to be elaborated on more too with Trevor's presentation after, but we are seeing these effects on the host immune responses just in when exposed to abiotic material. And so if it is some microbial level interaction happening between the environment and the host lung microbiome that exists already, we speculate that it could be that these microbes are literally like actually doing something, like their presence, or we have this other interest in whether or not it's a microbial exudate or if it's a microbial component that causes this irritation or level of respiratory inflammation.
Cool, thank you. Yeah, it's exciting to see how some of these pieces start to fit together. And on that note, we are going to move on. Thank you both for your presentation.